Okay, and we are back. It's Friday morning, April 9th. Uh, Chapter 28, lipids, we'll be finishing up today and starting metabolism. Remember, we said metabolism, glycolysis, citric acid cycle. We're just focusing on the organic reactions in isolation. We'll show you the overall schemes and put things in context, but for test 4 and what you need to know is how those reactions relate to things we've already covered, okay? So don't memorize the whole scheme unless you want to. Usually this time of the semester, that's a no-go, right? But some of you have seen it already in some other classes and maybe you're familiar with it. So, deal with it however you need to there. But there won't be a lot of it on test 4. It's just an added on thing. And also the final won't have any metabolism like that. That's really a topic in biochemistry. Homework 13 will be due Monday. That's for 28 here, the terpenes. And then quiz 10 is due Wednesday. And that's a good idea to see how we can ask these metabolism questions. Look at quiz 10 already. It's been posted for a while. So that will give you an indication there. Because these topics are not in our textbook, right? So you have to use the handouts and what we cover in class the next couple times. And then sample test 4, of course, is already posted. We'll be using that to review on Monday next week. And then I'm reserving, hopefully, next Wednesday for the final review. But you should be reviewing for that now because the final is over 351 and 352, right? Okay. Let's start up here, I guess. Sure. We left off last time talking about the fatty acids and, stearic acid and oleic acid. We've already learned about those and acyl triglycerides, the material in adipose tissue or fat cells. And we did the cell membrane components, the phosphatidylcholines. So I'd like to show a little bit about how they're made. This pathway is called fatty acid synthesis, and it's done by an enzyme called fatty acid synthase. And that's something about the name of enzymes, right? They end in ASE. We've learned about prefixes for different molecules for the naming, and enzymes, a lot of them are named ACEs, and it says something about what they do here. First, you take acetyl-CoA. We learned about that last time. It comes from pyruvate, undergoes that decarboxylation step. The enzyme has a base that can take a proton off one of the acetates and form an enolate. Now, we should know what enolates are by now, right? So they're anions at the alpha position. They're nucleophiles. And so the anion here can condense with a molecule of carbonate and lose hydroxide here, proton transfer, so water's coming off actually. I'm simplifying some of these mechanisms. And then there's a malonate intermediate. That's actually the resting state for this acetate anion. But we know malonates before, right? Those are the dicarboxylic acid, malonic acid that we've learned before. This is a malonate thioester, and ACP stands for acyl carrier protein. It's very similar to acetyl-CoA. It just has a bigger protein on it. It's also a sulfur ester, so don't need to worry about that. This can decarboxylate, lose CO2 back, and form the enolate, and that's the same thing up here, but now there's a different electrophile. It's another molecule of acetyl-CoA, okay, or ACP. Sorry, it's another thioester. So bring the electrons down, there's our electrophile, there's our tetrahedral intermediate. What reaction are we doing here? There's a name for this reaction, right? This tetrahedral intermediate breaks down, the thiol comes off as a leaving group, and we get this product right here, which is a beta keto ester with 2 more carbons on it. We've covered this name reaction before. What's the name again? Starts with a C, right? What is it? Who can? Claisen. Yeah, it's a Claisen reaction. Look at that. So this is just a Claisen reaction, beta keto ester. And here's our nucleophile, here's our electrophile, which is the same thing. It's just dimerizing, you can say, right? And it's forming now the 4-carbon ketone ester, okay? Now, it doesn't stay there. The fats don't have a beta keto group. The beta keto group gets reduced by a molecule of NADH. That's the reduced form of nicotinamide. The hydride comes off here, the pair of electrons on the nitrogen here, goes in, push the electrons up, and then protonate it. The enzyme has a proton source right there. And it gives what? The beta hydroxy ester product. Now that's an aldol type product, right? The beta alcohol ester. Now, initially, biochemists thought, well, maybe fatty acid is going through an aldol type process. No, then they found these beta keto intermediates, okay? And NADH is required. So it's not a direct aldol reaction because that would be what? Acetaldehyde as your electrophile, okay? Instead of the thioester. So it's two thioesters coming together doing a Claisen reaction. That's just like we've seen before, right? So there it is. The next step here, the fats don't have beta hydroxy groups either. The next step is deprotonation here. We can reform the enolate and then eliminate the alcohol. There's alcohol as a leaving group, right? And that's analogous to the aldol condensation reaction. So we've actually seen 3 reactions now that are really reviews of what we've covered already. The Claisen, the aldol itself, and then the aldol condensation. So look at that. It's right there to form the unsaturated ester product, alpha beta unsaturated, and that would be the result as if it were what? An aldol condensation. Now it doesn't stop there, but no, we need to ask ourselves, wait a minute, fats do have double bonds in them, right? Some fats are unsaturated or even polyunsaturated. So is this the spot where the alkene stays? Right there, and then the synthesis just continues? No, actually this is initially generated as a trans double bond, and it gets reduced in a 1,4 conjugate addition manner with more hydride. Another molecule of NADH reduces that, push the electrons the way they're shown here, and then protonate at that alpha position. There's the hydride that just came off of NADH, right? And then if you protonate this enolate intermediate, there we get to the fully saturated now butyrate or butanoate. 2 carbons now added. We went from what? Acetate down to butyrate, okay? And that should show you something. All of the naturally occurring fats are even numbered, right? 18, 16, 14, 12 carbons. And you can see here, they're units of the building block acetyl-CoA with 2 carbons. Now, what about this double bond? That's kind of a question hanging out there, isn't it? Let's see about that. Well, we can just continue with butyrate, go through the whole cycle here with another acetate. Here's our electrophile, here's our nucleophile, and go through that. Now we have a 6-carbon ketone, and then keep reducing. And now we have the hexanoate. So you see the pattern here? It's that same series of reactions, Claisen, then the reduction, elimination, and then more hydride. And then you go all the way up to the C18, C16, or C14, or even the C20, which is arachidonic acid, okay? So they all follow this pattern. It's a great pathway that leads to these. It's ultimately a reducing pathway because we're using NADH. We're reducing it, and these are hydrocarbons, right? These are mainly CH nonpolar bonds, so that would be a reducing pathway. But you see how we get to here? So I say etc., right? That same series of steps on this previous page need to be followed with the new electrophile each time, which is 2 carbons more, okay? Questions on any of that? All right, let's answer this unsaturated question here. Okay, so you can get to this point here, which is a C12 unit with a beta keto ester on it. And we can do the reduction and everything, the elimination. You see we get to here, the alpha beta unsaturated or enone form of the ester. Now that's actually the incorrect position because we know oleic acid, the monounsaturated one, is at the C9 position and it's a cis double bond, not a trans. So it's actually a little bit of a switcheroo here. At this intermediate, you deprotonate at the gamma position with a base, and you form the vinyl enolate. You can push the electrons all the way to the carbonyl. I'm not going to show that to you. And you can isomerize that and put the cis double bond right there as you deprotonate here at this gamma position. And then you protonate at the alpha position. You see that moves the double bond over. It deconjugates it to the carbonyl, okay? And it leaves it there. That's the 9 position, if you can see. And then what? We just keep adding 2 more carbon units a few more times here to get to the C18 oleic acid, which is a C9 cis double bond. Now, that's just acid-base chemistry and isomerization. So it's really not too much of a switch there. But there are enzymes that do that. It's called an isomerase in the fatty acid synthesis pathway. Okay. Questions on any of that? All right, we'll get into this, the cholesterol terpene pathway. And this will be our last topic here. But I think we need to go over to the board here, Jesharon, and look at a couple things first. We'll go back to those overheads in a second. But let's see if we can do this mechanism of the fatty acid synthesis together. How does this work? And again, it was identified what as this Claisen reaction, right? So let's see if we can follow this. So here's our 2 acetyl-CoA units. How do we get there? What's the first thing we need to do with this enzyme here? Fatty acid synthase. We have to turn one into a nucleophile and use the other as the electrophile, right? So what do we need to do right here? Let me help you out. What do we need to do to that proton? We need to do what to it? Take it off or deprotonate it. Okay, so yeah, we need a base here. And yeah, in your enzyme, you can use a base abbreviating the active site or the side chains here. And it's proximity issue, so let's go ahead and take that proton off. And I'm going to combine a couple steps here just to simplify things a little bit. So we'd have this enolate. We could draw the enolate, but let's just use the pair of electrons to go right here. So here's our nucleophile. Here's our electrophile. Okay, does that make sense? Can you draw the intermediate from that step? Let's see. That would be a tetrahedral intermediate, right? We'd have O minus, and we'd have thiol-CoA. Remember, that's a big molecule. Thank goodness for abbreviations, right? We only need to focus on what we're reacting here. And so we'd get this. Yeah, thiol-CoA. They're both sulfur esters. And then what do we need to do here to go from our tetrahedral intermediate to our product? This first step. Bring O minus down and kick off what? Yeah, kick off the thiol-CoA. Yeah. So you see, we regenerate that cofactor. And those are recycled in the cell. The cell makes very good use of a lot of these byproducts of certain steps. But that gives you that. And what's the name of this step? This whole reaction here that we just went through? That's a biological example of what reaction? Starts with a C, the Claisen reaction, right? So that kind of helps you out to see that, okay? And then the rest of the fatty acid thing is just reductions and eliminations. You can kind of see there. We could ask you about some individual steps there, but we give you the conditions, the starting material, and maybe the name of the enzyme and a cofactor, NADH in this case, those reducing things. So NADH has to reduce this to an alcohol, right? It's like a hydride coming on there. And we can eliminate the alcohol to an aldol type product. And yeah, we could go through the rest of the steps, but they're on the graphics. And all these graphics for metabolism and these steps here in this chapter as well are all on Learning Suite, okay? So you can look through this. All right, the last topic here are the terpenes. And this ties in with cholesterol also. These are C10 units that are mainly hydrocarbons or nonpolar bonds. Myrcene is a typical one here found in a lot of plants. You've probably smelled it before in bay plants or the bay leaves that are used in food preparation, whatever. Why do we call it a terpene? Well, it's made up of 10 carbons, and you can count them up here. And there's a lot of times this branching here. So here's a methyl group here. Here's an alkene group there. And you can kind of see the basic unit. If you break it up here, they're actually units of 2 C5 units. Now, a C10 unit is called a full terpene or a monoterpene. A 5 unit is called a hemiterpene. This is a so-called isoprene unit. These are also in plants. These are very volatile, though. They don't typically stick around, but the C10, and then there are multiples of that. There's C20, which are diterpenes, and we'll see even C30, which is a triterpene. They all come from this molecule right here. which is dimethyl allyl pyrophosphate. So I'm drawing out the whole two inorganic phosphates stuck together there. That's what the P superscript I stands for, inorganic phosphate. And the two here stands for pyrophosphate, that maybe you've heard about. That pyro has nothing to do with fires. It is an exothermic or an unstable bond. So yeah, it is energetic that way, but if you want to remember it that way, pyrophosphate, you probably heard about nothing. We saw triphosphates last time when we looked at ATP, right, adenosine triphosphate. But this is a hemiterpene, 1, 2, 3, 4, 5. And what you notice here, this branching at this position here, and it's kind of a gem dimethyl arrangement on the end there. That's considered to be the head portion of a hemiterpene. And this is the tail portion. And you can kind of look at a lot of these natural products and a lot of these type of lipids in plants especially, and we'll see cholesterol also. You can always kind of identify this tail and head position. The head position is closer to the branching, okay? There's more like substitution there in the carbon. The end position is always a primary methyl, okay? And let's see, this can be ionized, we say. There's a good leaving group, and we can get what? Dimethyl allyl cation, okay? This actually goes back to chapter 9, I think, in 351. We saw a lot about cations then, the SN1 type reaction where we're ionizing. This is a particularly stable cation, even though it looks like it's primary. Why? Because it's resonance stabilized, right? We could draw the tertiary carbocation here. So, you know, kind of review that. This one right now is primary. There you can go to tertiary. Well, how do we put these hemiterpenes or these C5 units together to make terpenes? Well, they all follow the same pattern. You can take an isomer here of dimethyl allyl pyrophosphate, where the double bond is just moved over here to this position. There's an isomerase that will do that. You can sometimes call this isoprene pyrophosphate in analogy to the simple hemiterpene that's has the phosphate off, or just eliminated right there. You can see how that could come about. So if that attacks there, what do we get? We need to draw an intermediate, I guess. We'd have our new bond here, and we'd have, if these electrons come out and attack there, we have a tertiary carbocation right there, right? And our pyrophosphate unit is there. And so how do we get to this one? Oh, geraniol pyrophosphate from geranium plants, okay? You've probably smelled those flowers, geraniums, right? Somebody's played in their grandma's garden maybe and smelled those. A lot of these terpenes are volatile enough to have a nice odor. Most of them are very pleasant odors. So how do we get from here, this intermediate, to here? Well, the enzyme has to eliminate that proton. You can do that easily right there and put your double bond right there. Now this is a full terpene, right? But it has the pyrophosphate that can do more reactions here, okay? So let's see, let's do maybe just a couple of these simple ones. I don't know, we got enough time. Let's see, maybe we'll just put this up as a challenge. Let's see if we can figure this out. Let's take geraniol pyrophosphate. And let's see, in conifer trees, pine trees, the main terpene is this. It's called pinene. Okay. And that looks kind of complicated, right? We've got a 4-member ring here. Here's the gem dimethyl here. If you count up all the carbons, it's still 10 carbons. How do we go from geraniol pyrophosphate to pinene in conifers? Or we could go to, in citrus plants, we could go to this, which is called limonene. For lemons, I guess, limonene. Okay, let's see, can you push electrons here? Here, I'll help you out to get started. Look at that. So if we do that, we'll get an allyl carbocation, right? So let's see. Okay. So you can kind of see, okay, we've got this methyl up here, and we've got to eventually cyclize. In both cases, we got to get to a 6-membered ring. So maybe let's do the limonene one first. Let's do a resonance structure of this guy first. What would we get if we push those electrons over there? See if we could get our carbocation there. And I'm kind of doing a couple things here. I'm resonating the electrons out here, a tertiary carbocation, and then we're rotating around this bond to put this in proximity, you could say, to this double bond up here. Now the enzyme does all this, okay? So this is bound in an active site. But all the intermediates, the electron pushing, we can do as organic chemists now, is the stuff we've learned. So yeah, here's our leaving group. Here's our allyl cation again. But now we've got to cyclize. What should we do next? Can you kind of see it here? What should we do? How about forming this bond right here? How can we form that? Let's see. Bring these electrons down. And what do I need to do with those electrons up at the top there? Just bring them down. Yeah. So let's see. So that would give this intermediate double bond over there. Oh, and that's right where we need it. And if we do that, oh, look, we got our carbocation up there now. Do you see how we could get limonene pretty easily? What do we need to do to complete the synthesis here? To get to, we need to put a double bond here. So we need to do something up here, right? What do you think the enzyme does now? It has a base in the active site. And that base just does what? Deprotonates again, okay? So it's pretty straightforward there. Yeah, and there we go, straight to limonene. Questions on that? So you can see a lot of these natural products, a lot of these essential oil compounds, including very complex ones, by the way, all kind of start out from this terpene-like framework, okay? Now, they don't keep all the carbons. They're not always multiples of 10, but a lot of them are, okay? So you can see a lot of them that way. And pinene, what would we have to do here? Well, we take this same intermediate, which would be this tertiary carbocation, and let's see, what would we have to do here? We have to get this 4-membered ring. 1, 2, 3, 4, right? So we need to take these electrons and cyclize right there. And we kind of have to pull this thing down, right? This isopropyl tertiary cation. Pull it down there, and let's see, we get that 4-membered ring forming. And again, I pulled this guy down here. And what do we get? We get the cation right there. And so how do we get to pinene? I think it's just one more step. And I think you can see that. It's just loss of that proton. Again, with a base in the active site. Okay. So terpenes aren't bad. All the steps go back to allyl cations, which is something we saw before in chapter 9. Okay. The electron pushing is pretty straightforward if we recognize the phosphate usually as just a simple leaving group. Okay. Questions on this? Know what a terpene is and know something about the basics of the mechanism. All right, let's finish off cholesterol here and see how much we need to do here. And this is just an extra little topic. We've already seen cholesterol as a component of the lipid bilayer membrane. It's made, though, from acetyl-CoA units. There it is, your universal guy, right? This ability to do the Claisen reaction and form these things. So let's see how we make these dimethyl allyl pyrophosphates and the isoprene pyrophosphate. That's what the key is here. And they're made in the liver. The body does make, higher animals do make cholesterol de novo in the liver, about 1 gram a day. In your body, you have like 400 grams of the material. It's 20% of all your lipid bilayer membrane, so it's quite a bit. But that's interstitial cholesterol, it's called. It's bound within the membrane. It's not serum cholesterol going through the blood. That needs to be transported because cholesterol is not water soluble. It has to be transported via an LDL, a lipoprotein, high density or low density. And maybe you've heard those terms before, and we'll kind of relate that at the end here. But here's how it's made. We form the beta keto guy, and then we have another enolate here, which adds as a nucleophile to that ketone to give the aldol product here. And this is called 3-hydroxy 3-methyl glutaryl-CoA. Glutaryl, well, that's from glutaric acid. That's one of the, oh my, such good apple pie dicarboxylic acids. There it is. One's a thioester here, and at the 3 position, you have a methyl and a hydroxyl. We just made this bond right here via an aldol reaction. We'll see that again in the citric acid cycle. We'll see the aldol reaction again, okay? And then the key step here is HMG-CoA reductase. HMG-CoA is short for this intermediate right here. This is the actual intermediate, the step that's inhibited by the cholesterol lowering drugs, the statins. Maybe you've heard of Lipitor or Mevacor, okay? Those statin drugs are real lifesavers in lowering serum cholesterol levels and lowering the risk of heart disease and cancer in many people. These are highly prescribed drugs right here. And what they do is they inhibit this pathway, which is the committing pathway in the liver to make cholesterol, okay? Now, it doesn't look anything like cholesterol yet. It's way upstream from it. But if you can inhibit this step right here, you can significantly lower the serum levels of cholesterol in the LDLs in particular. Anyway, so the step here where we phosphorylate, these are kinases where we use ATP to put on phosphates at these positions. And then what? It's a decarboxylation. Push the electrons here and then kick off that phosphate right there. Does the decarboxylation to put that double bond in place. And then there's an isomerase which just deprotonates here and then protonates over there to give this form of the isoprene, okay? And then here's the geranyl synthesis we just did on the board, okay? We put these together, allyl phosphate came off. Here's our allyl cation, attack there, and then what? Eliminate right there. And there's our geranyl pyrophosphate, okay? And we can do that again, add another one here. This is an enzyme called farnesyl synthase, which puts on the C15 now terpene, okay? So you see that's just another C5 unit coming on there. Now, this is kind of the key step called squalene synthase. And this is just for your information now. These get very complicated, these mechanisms. I just want you to appreciate these. But you can actually understand them based on all the principles we've covered in this class, okay? These cations here. This is unusual because it puts together this molecule called squalene, originally identified in sharks, by the way. If you're into your ichthyology at all, Squalus is the family of shark species. It's a tail-to-tail linkage here, which is unusual. It's harder to do. These steps here are well known now. Squalene synthase does this to put together that. This still doesn't look anything like cholesterol, does it? Cholesterol has 4 rings. Where are the rings? Okay, well, we're getting there. This enzyme right here, squalene monooxygenase, puts a epoxide on that one position there. Now we have something electrophilic. And look at this enzyme here called squalene oxidase. And then this enzyme right here will be called lanosterol synthase. This will take this squalene oxide and make this steroid-looking molecule. Now that's not cholesterol yet. That's C30. We'll have to remove 3 carbons here to finally get to cholesterol. How does it do that? This next reaction I've shown you has been called the most fantastic reaction known. 3 or 4 Nobel Prizes were awarded for this reaction right here. This was completely unknown till about 40, 50 years ago. And it took numerous biochemists and organic chemists to figure this out. That this one enzyme here takes squalene oxide, a little epoxide, just protonates the end epoxide. That allows the epoxide to open up and form the more stable tertiary carbocation. We've talked about that kind of thing, right? How epoxides react. Well, under acidic conditions, it's protonate, opens up there. And then if that becomes a tertiary carbocation, look, the enzyme orders the substrate so that it forms all the rings at the same time. Okay, now they're not perfectly synchronous. It is stepwise to some degree. But you have this ring forming right here. That's the A ring. And then you have this ring here, which will become the B ring. And then you have this next ring here. And these are all just cations that are formed from a cascade type reaction, called a cation-olefin cascade reaction. And you get to here. Now it kind of stalls at this point, and then there's a series of hydride shifts and even methyl shifts all across the framework here. And then it gives this molecule right here, which is lanosterol. Now, I've never put that on a quiz or a test, okay? So I don't study that, but just appreciate it at least and be in awe of Mother Nature and how she puts together these fantastic molecules called steroids, right? So the main one is cholesterol. That's the dominant one in the body. It's not a hormone. Not all steroids are hormones. Cholesterol is not. It's not a signaling molecule. It's just in the membrane. And cholic acid is not a hormone either. This is then produced by the gallbladder to be an emulsifier for food material passing through the GI tract. If you don't have this, you have a lot of problems digesting non-water soluble materials in food, mainly fat that you eat. And so cholic acid you see is emulsifier because it has a polar part. It is the salt form of this carboxylic acid. You can see some nonpolar parts to the molecule, which would help emulsify or bring those fats into contact with water. Now, from cholesterol, the ovaries and the adrenal glands make progesterone. Progesterone in females helps control the pregnancy process. But look, it's just a tiny amount, 5 nanograms per milliliter. Okay? And yes, guys, males do produce some progesterone in the adrenal glands. Okay? It's a precursor to all the other hormones. Progesterone's kind of a key one. And what it is, it's a methyl ketone on the D ring. So this hydrocarbon nonpolar part's chopped off there. Now, that's not one I expect you to know, but you see the enone already in progesterone? That holds true into testosterone, which is just an alcohol in the D ring. So you should recognize testosterone as this enone right here. And this is responsible for muscle growth and other things in males in particular. But females also have this to control different processes in their growth events as well. Now, in females, estrogen is made, which is the aromatic D ring, the phenol. And that's about all I expect you to know. There's other ones like cortisone, cortisol that you'll learn about in medical school that control inflammation and metabolism. Some of these other ones have very specialized things. And we do teach classes here at BYU, endocrinology, where you go into a lot more detail of these. Yes, a question?
So you said that cholesterol is not a hormone.
Right?
The rest of the hormones are also steroids?
Yes. There you go. Yeah. All the steroid hormones all have this 4-ring structure that's in common. But a hormone means it's made in one part of the body and then it's transported in another part of the body where it signals a certain event. And these are the events that they all signal, okay? But yeah, okay. Yeah, that's all we need to. Synthetic steroids are not going into this. Anabolic steroids have been abused, although there are some applications of them. Some of you have probably taken prednisone for allergies or asthma, which controls inflammation in the lungs. Some of the others are lipoproteins, LDLs, HDLs. Maybe you've heard about those. Here's the bad ones. The LDLs are the low density, bigger ones that can cause occlusions in the arteries or atherosclerotic plaques, which can lead to heart attacks. Okay, so these have too much fat in them. These are transport vesicles, right? So the cholesterol component in the LDLs can be very high. Why do you need the transport? Because cholesterol is not water soluble. It has to be transported through a vesicle through the blood. Okay, and this transport can get out of whack there. The good ones are the higher density ones. They're smaller. They have less cholesterol, but you can look at that. They also transfer triglycerides, which are the adipose tissue fats. Those are not water soluble also. And here's the statins. You can kind of see their structures here. They're beta hydroxy esters, which is what the substrate here, HMG-CoA, is for making the isoprene in the liver. So if you shut down the synthesis, and you see this part of the molecule is the same here in Lipitor. Okay, this is a multi-billion-dollar a year drug, Lipitor, right here, and it's because it just mimics this part of that substrate right there. Anyway. Okay, that's probably enough. And it was designed based on rational drug design. We've already mentioned that. Okay, and just know the basics of that. Like I said, just be able to tell testosterone apart from estrogen. There might be a general question like that, but certainly not that whole mechanism and everything. All right, let's get into glycolysis and metabolism now. Okay. need to talk about a few things, and we'll go through you know, many of the key reactions here, not all of them. I'll just relate them to reactions and steps we've already covered. You're not going to reproduce the whole schemes, but be aware of what the general process is. We're converting glucose into CO2 and water, and we're making energy in the process. These are exothermic reactions. The energy is harvested or converted into ATP, which is the energy molecule within cells. Glucose is the energy molecule coursing through your veins. It's blood sugar. It's taken up by all cells in the body, but it's not used as energy within your cells. It's converted into ATP. So it's kind of a progression here of the ideas. But this is how we stay alive, okay? Body heat is based on these exothermic reactions. It's not all converted into ATP here at the bottom. But glycolysis, first step, I've already mentioned that. That's glucose to pyruvate. Pyruvate to acetyl-CoA. This is all in the cytosol of the cell, if you remember a little bit about your cell anatomy there. And then acetyl-CoA, the citric acid cycles in the inner membrane of what organelle? Let's see if you remember a little of this. Mitochondria, right. They're the energy powerhouse of the cell, and that's because they make ATP. Well, where's ATP? We haven't seen it yet here. Oh, we see CO2. There's one of the combustion products of burning molecules, right? It's the same thing. These are the same byproducts of combustion, of hydrocarbons or whatever we're burning, CO2 and water ultimately, there it is. The reduced cofactors here, after they oxidize glucose to CO2, then the reduced cofactors, NADH, FADH2, reduce water, or oxygen, sorry. So this is the aerobic part of metabolism. Up here is the anaerobic part. Maybe you've heard about that before. You'll see how the intermediates are, and then we get to water and then ATP, okay? And so let's look at it. Important cofactors, we've already seen a couple here. Acetyl-CoA and NAD+, the main one. FAD is also in here. Here's glycolysis, 10 steps. Wow, end of the semester. Why am I going over this? Well, it is glucose. So let's see, the first step is just putting on a phosphate. Okay, so far so good. And it uses ATP. Okay, this is the investment stage of glycolysis. We have to use some high energy molecules, put it on. There is an enzyme involved, hexokinase. Kinase means enzymes that put on or transfer phosphate groups, okay? Maybe you've heard about that too. It is exothermic because pyrophosphate triphosphates are releasing that energy. They're anhydrides of phosphoric acid. So a lot of like charge repulsion goes on there to form 6-phospho glucose, okay? And then glucose here is isomerized into fructose. Wait a minute, we go from the aldose, and there it is, the hemiacetal. That can open up to the aldehyde form, and you can draw the Fischer projection here and have the phosphate at the 6th position. And then you isomerize to fructose, and I showed you that isomerization process. That enzyme is called isomerase, okay? Yeah, question. They have to be, no, that's in equilibrium with beta. So I'm just showing alpha right now. Yeah, so these are hemiacetal anomers, so they're in equilibrium with their open one. And same thing here, okay? The fructose can be alpha or beta. We're showing alpha right now, okay? And then we put on another phosphate group, another fructokinase, fructo referring to the substrate fructose, and we use another molecule ATP to form the diphosphate. Now, we do a reaction with aldolase. Aldol, that's a reaction we know, and I've just mentioned it. Aldol puts on a bond, makes a carbon-carbon bond. This is actually a retroaldol reaction, which breaks diphosphofructose into 2 small molecules, which stands for DAP, or dihydroxyacetone phosphate. And there's your acetone, that ketone right there. And that ketone right here came from fructose. There's the ketone right there. And we'll go to the board and go through this reaction. And then the other byproduct here of this retroaldol is GAP. GAP stands for what? Glyceraldehyde phosphate. GAP and DAP. And it's okay to use those acronyms, okay? Biologists do, and biochemists, and so we can too, GAP and DAP, but make sure you know what they are. DAP can be isomerized, or you can take the carbonyl here and isomerize it over to the aldehyde position, just like this isomerase, okay? It goes through that enolization and then tautomerizes, you protonate on the other side. We went through that mechanism last time. You can review that. It's the same step there. And then we'll oxidize this aldehyde. Notice DAP is converted into GAP. So for each molecule of glucose, you generate 2 GAPs, okay? So from here on, it's doubled the whole pathway. And then we oxidize to the glycerate, which is a carboxylic acid anhydride with phosphate on it. We can then transfer that phosphate to ADP and make a molecule ATP. There's the first payoff. We're making ATP now. And then we isomerize. The phosphate goes on the 2 position, and then we eliminate water right there, or enolize. That's what the enzyme means, enolase. We do the elimination there to form the phosphoenol pyruvate, it's called, or PEP. And that's a good acronym. It is a peppy molecule because it can transfer this phosphate on the enol to ADP and make ATP and make ultimately what? Pyruvate. Okay. So that's not the whole story. That's just the cytosol events. Okay. Let's do a couple on the board here, see how many we can do. I'm not doing them all. Let's just go right to the aldol one. Okay. The aldol one's kind of the key one here. Let's see. So what do we have? We have phosphofructose, and we need to draw the fructose molecule, right? So we should know what fructose is. And we're going to draw the Fisher projection here. And remember, it is left, right, right. And it is related to glucose, of course. But now we got the 2 phosphates on there. Now, aldol reactions make carbon-carbon bonds. Here we're breaking a carbon. How can we do that under these mild conditions? That's like 90 kcals per mole. But look, if we have these 2 phosphates on there, and by the way, what's the charge on a phosphate? 2 minus. This molecule overall is 4 minus. And those are like charges. That's highly repulsive. This is a high energy molecule. How can it minimize that like charge repulsion? It can break apart. Create the 2 molecules, GAP and DAP, that only have the 2 minus now. So part of this phosphorylation is to tag that molecule to keep glucose in the cell. It can't be transported back out. Once it comes in and gets phosphorylated, it stays in the cell because it's charged now. It can't get through the membrane, which is a nonpolar region. Okay. And so how do we break this bond here? Let's see what the enzyme does. Well, it has a base here. This is aldolase, right? And it's the name of the enzyme. And so let's see. The bond we need to break is right here, right? And how are we going to break that? Well, you see here's our ketone, and at the beta position to that ketone is this position at C4, okay? That's where the aldol is going to operate, okay? So let's see. If we can take this proton off here and then push these electrons down here, we can push these electrons here and then up onto that position. And there's a protonated base that can take that O- charge and get protonated there. So there's a free base here and a protonated base there, a proton source, you could say. So what does this go to? Let's see. We've got, phosphate and enol now. Yeah. And that alcohol at the 3 position is still there. And what happened at the 4 position? Well, yeah, that becomes, look, that becomes, that is glyceraldehyde phosphate, or GAP, okay? And we broke this bond right here. See, it's not there anymore. That's this pair of electrons coming out and flowing up to the protonated carbonyl. So it's not too mysterious. And then how does this work here? Well, this isn't quite DAP yet, right? We need to take our base here and take this proton off, liberate the electron. And what? We need to protonate right there. And there you see we'd get what, the ketone there, phosphate there, and what, just free alcohol right here. We can rotate around that bond now. Okay, so this is an enol. The alkene alcohol is on there. And you can see that this would be DAP. Okay. Now let's look at the aldol in the forward direction, because that's how we're used to seeing it. And in fact, there's a process in cells called gluconeogenesis. What do you think that stands for? Gluconeogenesis? Making glucose de novo, or of new. And if an organism needs glucose, you can actually make it from GAP and DAP, which can actually come from other metabolites in the body, including amino acids or fats. So there's a lot of redundancy here in these metabolism pathways. We're looking at it in the forward direction of glucose on down. But there's actually other intermediates that can interfere in other processes. And this gluconeogenesis pathway going in the opposite direction is also controlled by aldolase. Okay. And so how would it work? Well, you take off the proton here, you'd flow it down here, and then you'd protonate right there, right? And look, that would make the bond, and that is a true aldol reaction now. Okay. This here you'd call a retroaldol. Okay. But it's controlled by the same enzyme. So do you see that, how that can work there? And GAP and DAP are kind of the key thing here. And you can also isomerize this one, and that's that fifth step where you isomerize DAP into GAP. So then you wind up with two GAP molecules, okay? And then we continue on. Let's see, which other one did I want to do here? Yeah, that isomerase is not a big deal. Oh, let's look at the glyceraldehyde 3-phosphate dehydrogenase, converting GAP into glycerate. So this is kind of a key one here. Let's see, if we have glyceraldehyde and we got the phosphate on there and the OH, okay, how do we go from that to this, which is now a carboxylic acid with a phosphate on it? And, well, two phosphates, right? And this, okay. Now there are a couple cofactors here, NAD plus, okay, which makes sense. We need to oxidize this aldehyde to the carboxylic acid. And we also have inorganic phosphate here, okay? So let's see, inorganic phosphate. What do we got? Let's go ahead and attack here and see what we get. Well, we get this. And what? We already have a phosphate on there. So how are we going to do this oxidation? Notice this here, you'd say is like what? A hemiacetal, right? Well, we need to get this hydrogen off with its pair of electrons. That was our aldehyde hydrogen. Well, how do we do that? Well, here's NAD plus. And make sure you can draw NAD plus and NADH. You may need those, and those are the very common oxidation reactions. So knowing NAD plus is kind of a key here. And so what can we do here? We got the plus charge right on that nitrogen. So our enzyme will take this hydrogen off, liberate these electrons, and look, hydride can go in and reduce the NAD plus. And look, that goes to the glycerate now. Goes directly to it. And what's the byproduct here? NAD becomes NADH. Okay. So hopefully that makes some sense there on that. I don't think I'll go through the rest of the steps here on glycolysis. The others I've already described, I think. We've got a mutase, which transfers this phosphate to that position. And you get the glycerate. This phosphate here gets transferred to a molecule of ADP. ADP is the adenosine diphosphate. That can grab that phosphate, you see, and form the glycerate here. And then this isomerase just puts the phosphate at the 2 position. And then you see, you can just eliminate water right here. So how does that work? Well, you need to protonate your alcohol with a protonated base, and then also in the enzyme here for that step, take that proton off, and you see that gives you the phosphoenol pyruvate. Okay. So yeah, I guess I am summarizing everything you need to know here. And this will also transfer a molecule to adenosine diphosphate. That phosphate can come off the PEP position. This is that molecule called PEP I just mentioned. That can come off, you see, and then in the enzyme, what do you have? You just have a protonated base. And that gives you the methyl group and the ketone and the carboxylate. So there's pyruvate. Okay, so they're all reactions we've seen before. This is just an elimination, a dehydration, right? And this is just, transferring phosphate again. And this is an enolate that we're just protonating, okay? So hopefully that makes some sense, chemically at least. We're not going into all the control elements and all that and all of the energy values for each step. You'll do that in biochem later. But we're just focused on individual steps, okay? Given a starting material, the name of an enzyme, can you come up with that product in an individual manner? And the key one, I think, in this case is the aldol reaction. Aldol, the retro aldol. Okay, I think that's enough for today. If you've got questions on this we'll do citric acid cycle on Monday, just a little bit. And I think we're pretty much done there. And then we'll be reviewing for test 4 on Monday also. Okay, but read ahead, go through the citric acid cycle before Monday to look at that. And all these graphics again are on Learning Suite there. So very good, guys. We'll see you next week. Thank you.